All right, welcome back. I decided I am going to split these last two parts into their own videos. So in this video, we're just going to be focused on the pox virus replication cycle. And unfortunately, pox virus replication is fairly complicated, but we're just going to do the most basic overview possible here. And so just to kind of lay the foundation here, all the orthopox viruses are enveloped and they're what are called brick-shaped viruses for obvious reasons here. These are huge viruses, so you can actually see the virus particles with a decently powerful light microscope. It's got a huge double-stranded DNA genome. You have these really big, what are called the inverted terminal repeats here at the either end of the genome, and then what's called the central conserved region in the middle, which encodes, about 200 genes, which is kind of insane for a virus. Another interesting feature of the genome here is that the ends are folded back on each other in hairpin loops. So you basically have a covalently closed double-stranded DNA molecule instead of the ends of the DNA being open as they normally would be. Um, and just in terms of the overall replication, this one's kind of unique for a double-stranded DNA virus because it replicates completely in the cytoplasm, so it doesn't enter the nucleus at all, as most DNA viruses would do. And the fact that it does that probably explains why it has so many genes, because instead of using some of those host genes that would be in the nucleus, it's bringing its own and making its own proteins to complete replication. The virion itself is pretty complex. I think I may have showed you a picture of these in an earlier chapter. But you've got the viral DNA here in the middle, and it's actually associated with a number of different proteins that package it up into, what you would normally see in a eukaryotic cell, almost like chromatin. You have this interesting kind of dumbbell-shaped core, they call it, which is two layers of these T-shaped proteins. So you could think of that as essentially kind of the capsid protein there. But you have these kind of reverse-facing T-shaped proteins that make this protective layer around the nucleic acid. The outer layer, they're calling the palisade layer here. Um, And you've got a bunch of proteins inside the core that aren't being shown here, but you've got an RNA polymerase. You've got a bunch of different enzymes that are important for transcription and messenger RNA processing. So there's a lot going on inside that core that you can't really see in this cartoon. It would make it too busy. And then the core is, so this is enveloped, the core is surrounded by a membrane, and you can see there's a bunch of proteins associated with the surface of the membrane. But then you also get these structures called the lateral bodies, which are basically just kind of the void space there on either side of the skinny part of the dumbbell inside that membrane. So we get two lateral bodies. And because you've got all these protein protrusions that the membrane covers here on the surface, it almost looks like if you're looking at corrugated cardboard or something from the side. So they call it a corrugated appearance. Um, and really have two different infectious forms here. We've got what's called the mature virion, which is shown up top here, um, which is basically what we've been talking about up to this point. But then you also have what's called the enveloped virion, which has an additional envelope with a bunch of associated proteins as well. Um, so, the basic internal structure is the same, but you will see both of these in a pox virus infection. And we've got, as I said, a bunch of proteins, at least 20 that are associated with that mature virion envelope. So kind of the yellow protrusions here, and we've got at least 29 associated with just or sorry, I said that backwards, 20 associated with the mature virion, and then another 9, 29 associated with that enveloped virion overall. So uh, pretty busy at the surface of the virus particle. Um, so what's the difference here? Well, the mature virions tend to be more stable. Those are typically going to be what would be passaging smallpox virus between two different hosts. Whereas the envelope virions, that outer envelope is pretty fragile. So you're usually just going to see this within an infected host, helping facilitate cell-to-cell spread of the virus within a single host. So that's seems to be the functional difference between those two. And just keep in mind, pretty much everything we know about this replication cycle is really coming from research with vaccinia virus. So if you really wanted to work straight up with variola, right, smallpox causing virus in a lab setting, there's really only two places in the world you could do that, at the CDC in Atlanta or this place called Vector in Siberia. Sounds like a Bond villain or something, Vector. So there's not a lot of variola virus research going on, but there is a lot of vaccinia virus because it's not going to kill people if it gets out. So again, most of what we know here is based off of vaccinia virus replication, but we believe it to be true for variola as well. All right, so let's look at attachment and entry here for just a minute. So here we're looking at that doubly enveloped virus here, and there's a couple of proteins that are known to be specific for binding to the cell surface, and basically they will initially bind, and then that will trigger that outer envelope to dissolve, and then other proteins on the surface of the mature virion will bind and will actually get uncoating and penetration at that point. And you'll get this big entry fusion complex that you'll see on the next slide that's got at least 11 proteins in it. So there's a lot of proteins involved with this entry process. But just to kind of give this the simple version here, so that mature virion, you've got proteins on the cell surface that'll associate with these two glycosaminoglycans, chondroitin sulfate and or heparin sulfate. And then there's also some proteins on the virion surface that'll bind to laminin, which is a common component of animal cell extracellular matrix. So that's kind of the initial binding. And then really the specific protein receptor on the cell surface is this one called MARCO, which stands for macrophage receptor with collagenous structure. We're not going to go into the collagenous part of that. But once that is bound, that's going to trigger the assembly of this whole fusion complex here, all the purple proteins you can see in that cartoon. And that's going to mediate basically fusion of the viral envelope with the plasma membrane, and is going to allow that dumbbell-shaped core to enter the cytoplasm of the cell. And so even though we said poxvirus does not need to enter the nucleus, it does immediately head toward the nucleus. So it'll get transported, that dumbbell-shaped core, on a network of microtubules to basically the surface of the nucleus, where it will remodel some of the endoplasmic reticulum to create little membrane-enclosed viral replication factories, essentially. And you can see that in this image here. So this is a fluorescent light microscopy picture where they have the nucleus labeled with this stain called DAPI. So the blue here is the nucleus. And then they have this viral G8 protein in the fluorescent green here. And you can see it's basically just sitting there right at the surface of the nucleus. All right, so the virus, we said, is bringing a lot of proteins with it, so it's got all the enzymes it needs to start transcription, processing the mRNAs that are produced. It does not have to go into the nucleus to get that done. And it's going to use an RNA polymerase for that, obviously. It's going to add the 5' cap, poly-A tail, everything that you would need to have functional messenger RNAs, which could be translated. And this is going to start quickly. So all this, transportation to the nucleus, remodeling the membrane, actually starting the process of transcription is happening within 20 minutes. So it's quick. And like we see with many viruses, we get these waves of transcription. So we get these early genes transcribed within the first hour and a half. And these are all going to be genes coding for proteins that are needed for replication. Okay, and I'll show you replication here in just a second. Um, some of those then are going to activate transcription of what are called the intermediate genes. These is basically just another wave of transcription factors, which would then eventually activate the transcription of the late genes. So we get this separation of almost 3 hours here, or 2.5 hours between the early and the late. The late genes are making all the kind of structural proteins, right, that we would need to actually assemble the virions. And just as we talked about with other viruses, the whole purpose of this kind of scheme, the waves of transcription, is to make sure that the virus is giving itself time to replicate its DNA before it starts producing the structural proteins that would package up new virions, because it doesn't want to run the risk of packaging up empty virions with no genetic material. All right, so what does the DNA replication part of this actually look like? And it's not too bad. It's actually kind of a cool system. Um, so remember, we had this covalently closed double-stranded DNA. So the first thing we do is we make a nick near the end of one of those strands. That will allow this end to unfold and linearize there, so you get this overhang. Um, the DNA polymerase can now extend from the 3' end here, so it starts elongating, and because there are complementary sequences here in this overhang, that's going to tend to fold back on itself and form what's called a little hairpin loop. As it folds back, now the DNA polymerase is pointed back towards the double-stranded DNA here, and it will move through unzipping and replicating as it goes. You'll notice the other overhang also forms a hairpin loop. So we basically replicate all the way back through this double-stranded DNA until we come to the second hairpin there. And now we basically have two complete copies of the genome that are still attached to each other. So we'll use an enzyme to cleave that in half, and now we've got our two copies. So lather, rinse, repeat there. We make a bunch of copies of the genome. Now we're ready for potentially for assembly and release. And remember, all the replication was happening in this virus factory, kind of right at the surface of the nucleus. So in this picture, you've got the nucleus at the bottom, cytoplasm in yellow, outside of the cell up here in white. And here we are in this little virus factory. So we'll have these what are called little crescent membranes. So these are little pieces of membrane that were basically stolen from the ER, and they're kind of shaped by this scaffolding protein called D13, which are the green little hexagons here in this cartoon. Then these crescent-shaped membranes are attracting some of the viral proteins that were being made, as well as the viral DNA, and it will eventually completely enclose all the necessary proteins and the nucleic acid, and we'll seal it up and form what's called the immature virion, which is shown here. Okay. So we already have this now essentially membrane enclosed mix of proteins and our viral DNA. That's going to bud out of the viral factory, um, picking up a membrane there. That the scaffolding protein was obviously removed. Forgot to mention that. But some of the proteins that are enclosed here are going to help with the formation of that dumbbell-shaped core that we already talked about. And at this point now we're out in the cytoplasm. And this is really the mature virion now, and those mature virions would be released when the cell ruptures, right? So we're basically filling the cell with virus to the point where it's going to burst and lyse. What about the envelope virions? Remember, the envelope virions were the ones that I mentioned were primarily responsible for cell-to-cell infection inside an individual. So some of those mature virions are going to bud through the Golgi and pick up that second membrane. So and as they bud, so they bud in, they pick up the second membrane that you would find on the surface of the envelope virion. As they bud out, they pick up another membrane, so we get what's called the wrapped virion. But that outer membrane then would be lost upon exocytosis, fusion with the plasma membrane. Um, and there's kind of this interesting phenomenon that happens with these enveloped virions. So as they exocytose, you'll get this cytoplasmic protrusion with these kind of actin tails, they're called, so cytoskeletal tails. And it's almost like the cell is giving that virion like a little kick in its butt as it leaves the cell, so kind of launching it off and making sure that it moves on as it leaves. Um, yeah, I think I said all that. All right, I'm going to stop there and I'll pick up in the last part on eradication of smallpox.